Hello all, welcome back to the BioRocks channel. In this video today, we'll be learning about the plasma membrane. Okay, so the membrane, it might be the plasma membrane covering up, lining up your cell, or it might be the membrane which is lining up your organelles. They are one in the same in the structure. So today we are going to deal with that particular thing, that is about the membrane, okay, of your cell as well as the cell organelles, that is your plasma membrane. So children, if you're going to look at the plasma membrane, it can be easily described as the what we call as a fluid mosaic model, because mosaic is nothing but it is just being interrupted or with many of the different sorts of proteins here. Like you can see like a peripheral proteins, the integral membrane proteins we have the transport proteins, we have cholesterol in between, we have these carbohydrate chains coming up attached to the lipids, so we call it as glycolipids, and we have the carbohydrate chains attached with the proteins, what we call as glycoproteins. So these all in between of the bilipid layer, so we call these all the mixture of things togetherly as mosaic as well as the fluid, because these molecules are going to show the movement to the sideways and towards any of the directions, like so they are having a fluidity. So we are going to call this as a fluid mosaic model. So let me get into the details of this fluid mosaic model and introduce you with all the sorts of the proteins, embedded proteins, and their functionalities. So children, now I'll be starting up with your bilipid layer. So if you remember in the last few videos, I was talking about the bilipid layer, telling that these are going to actually have what we call as hydrophilic heads, as well as it's going to have these tails. Okay, so we are going to have these as the main constituents. One is going to be your hydrophilic heads with the tails here. Okay, so the ones here, what I'm talking about, this particular thing is going to be hydrophilic, and the tails are going to be hydrophobic. This actually is comprising of a phosphate. I'll show you these, which gives a charge for it, which makes it polar, due to which only polar molecules through, can pass through it, can pass through it. And these hydrophilic molecules are the polar ones, okay? And remember children, the hydrophobic parts are made up of two chains. One is going to be a saturated chain, fatty acid chain, and another thing is going to be a unsaturated fatty acid chain, because it has a double bond in between the carbons. So we have a bilipid layer, because this is the first, this is the second. So the bilipid, phospholipid we call it as the phospholipid bilayer. Okay, this is phospholipid because of the presence of the phosphate groups, and the tails, the fatty acid tails that we are going to have. This is a hydrophilic ends. These are polar, so they are very friendly with the water. So that's the reason we call them as hydrophilic in nature. Okay, so this can be your extracellular fluid, children. Extracellular fluid towards the ECF, that is your tissue fluids, and this is facing towards your intracellular fluid, which is none other than your cytoplasm, towards your cytoplasm. So your plasma membrane is a biphospholipid layer, having the first and the second layers made up of the hydrophilic heads and hydrophobic tails they are having. And these hydrophilic ends are towards the extracellular fluid, or what we call as the tissue fluid. And the other end is towards the intracellular fluid, that is cytoplasm, which comprises of 80% of the water. So they are facing towards the water, and this is towards your tissue fluid which is being present. So children, what I'll do is let me show you this particular hydrophilic and the hydrophobic tails so that I can clear your concept here and then can move forward into the further set of topics of the phospholipids. So I was talking about this particular thing where I was talking about the two, there is a biphospholipid layer where you can see that these are the heads, which are the hydrophilic heads, and these are the tails. Each head is having two tails, one of the saturated fatty acid and the other is going to be unsaturated. So it's going to be bilipid layer where this is facing towards extracellular fluids and one is going to face towards intracellular fluid, that is going to be your cytoplasm, that is intracellular fluids. Now coming on to the one particular phospholipid, if you're going to see, so this is your hydrophilic head, which is going to have a phosphate having an negative charge, which makes it polar molecule with a glycerol also in it. So this is going to be your hydrophilic head. And this is the saturated fatty acid having only the single bonds, whereas this is the unsaturated fatty acid having a double bond between the carbon and carbon. And this is going to be hydrophobic because these are the fats. So these are lipids. So these are hydrophobic and this is going to be the hydrophilic. Okay, so now children, what I will try to show you is all the other embedded things in between these bilipid layer, as I told you, because it's a mosaic model having the glycolipids, the glycoproteins, and possessing the peripheral proteins, okay, having these transport proteins, the ion channels, okay, and in between we have these cholesterol molecules. So we will speak all about, so just remember these all are the things which are making your bilipid layer as a mosaic model because of all these mixtures, children. So now let me come into the details of this. Now I'll be entering from one to the other. So the first thing that you can see as mosaic model. So since we are calling this as a fluid mosaic model, so mosaic model itself is telling that it has a mixture of many things. Mixture of many things, children. So what are these many things here are? It might be the presence of cholesterol, cholesterol. We may even have many number of embedded proteins here. There are different sorts of proteins, very specific, very different from each other. Togetherly, we call them as embedded proteins, which is making the plasma membrane, making our plasma membrane as what? As a mosaic model. So children, let me first come to the topic of cholesterol here. Cholesterol. So what cholesterol does is this is going to actually decrease the fluid, what you call it as a fluidity of our plasma membrane, of our membrane. So what it brings is it brings a rigidity. So this is bringing a rigidity into our membrane, children, our cholesterol. And remember that at high temperatures, if at all, if you see a condition that if there is no cholesterol, if at all, if there is no cholesterol in the plasma membrane, then what would happen is at high temperatures the fluidity of your plasma membrane would have been more. If there is more fluidity, children, there would be more water seeping, as well as water soluble things, water soluble things moving out and in, right? Moving out and in of your cell. So imagine if the water is moving out of the cell, this would have led to the dehydration process, but we are trying to avoid the dehydration just because luckily we have the presence of this cholesterol inside the plasma membrane, which is increasing the rigidity, which is decreasing the fluidity, by which at even high temperatures we are trying to, the cholesterol is going to try to, if there is cholesterol, it's going to what we call is avoid the leakage of water molecules from your cell, leakage of water molecules from the cell. So this is one of the finest functions, molecules from the cell of your cholesterol. So this is most very important to remember that even cholesterol has a great functionalities like giving rigidity, and it is nothing but, you can even define it as, it is restricting the lateral movements. It is restricting the lateral movements in the plasma membrane. It's providing the rigidity to the cell. Okay, this is very important, children. Next, I'll be talking about the embedded proteins. There are different types of embedded proteins. The embedded proteins are of different types here, children. So amongst them, what I would like to cover up is going to be the ion channels. First, let us see about the ion channels, children. So if you remember, in the videos that I have made about the transport of molecules across the cell membrane, I was talking about these ion channels. If you remember, I was talking about the sodium potassium pump, right? I was talking about the sodium potassium pump. So these ions like sodium ions, the potassium ions, the chloride ions, the calcium ions, all these ions. What are the ions? Are nothing but electrochemically or electrically charged. These all are the electrically charged atoms, right? So these ions are very, very small. All these ions are of very small sized. They can easily diffuse out, okay, without any investment of the ATP. They can easily actually move or diffuse in and out. To transfer them, we have some special proteins here, children, and these special proteins are being called as ion channels. We are going to call them as ion channels, we are going to call. So these ion channels are going to let the, what we call as a diffusion of these ions across the cell membrane, and they are just being diffused in and out of these. Okay, these are the ion channels, and remember that every ion has its own specific channel. So there are specific ion channels, there are specific ion channels for the specific ions. Like calcium has its own type of ion channel, sodium and potassium are having their own sort of ion channels, and the chlorine has its own ion channels. But remember, children, that ion channels are actually closed. They open out when the ions have to be pumped. So they can close as well as they can open based on their transportation process. So this is about the ion channels. And next, coming on to, we have another one, children, that is going to be called as your transport proteins. So this also I have specified in the videos where I made about the movement of materials, molecules across the cell membrane. So we have the transporter proteins also. These are the different types of proteins again. And if you speak about the transport proteins, children, they are going to transport only the large sized molecules, okay? Large sized molecules they are going to transfer. If you remember, children, I was talking about the facilitated diffusion, where the diffusion is not across a membrane freely, but they are being facilitated by some transport proteins, and there is no investment of ATP here. And such a molecule that is being transported by facilitated diffusion, the best examples I gave you was about the glucose molecules. But children, in order to transport your amino acids, what we need to do is we need to invest the ATP molecules using the transport proteins only. So we call this as a active transport, very much different from your facilitated diffusion, because in facilitated diffusion there is no usage of ATP, while in the transport of the amino acids we require ATP. Remember children, why am I speaking about the large size? Because when you speak about the glucose molecule C6H12O6, almost you are passing 24 atoms through the transport protein. So we use this transport proteins but cannot be easily diffused. They can never be easily diffused. So since because of their huge size we are using these what we call as transport proteins we are going to use. I hope you understood. And even the protein structure is very complex because of the amino acids. They are very large than the glucoses, and to move them we require even transport proteins as well as ATP molecules also. So that is a thing that you need to remember for the transport proteins, children. And in the next coming thing that we are going to see, the embedded proteins are going to be your enzymes. So there are many sorts of enzymes facing towards the bilipid layer, towards your cytoplasm. And there are even enzymes facing towards your extracellular fluid also. We all know that these are the enzymes, are the proteins which are catalyzing your several biochemical reactions inside the cell. Several biochemical reactions have been catalyzed, children. So these are the enzymes, which are the proteins which are catalyzing your biochemical reactions. So these are being present here on what we call as your cell membrane. See, for instance, if I draw a cell, for instance, if this is going to be your cell, so 80% of the cell is going to have water, that is intracellular fluid, which is going to be your cytoplasm, and this is going to be your tissue fluid. And if you look at all these proteins, how they are present is they are present above as here, like these are the what we call is the proteins, which are the embedded proteins. But remember children, there is one set of ion channel which is going to be present all through the length. So that is going to be this, and these are going to be what, these are the actual ion channels what I was talking about. These are the ion channels. The basic difference is ion channels are very long and they're running across the bilipid layer. But if you look at this particular other proteins which are being present, they are only present embedded in the extra, that is your what you call is outer membrane. So ion channels are running in between the whole togetherly between your what we call as the, let me choose, yeah, so they are running in between both of the bilipid layer here. But whereas many of the other proteins that you are going to see, the embedded proteins, are just present towards the outer surface here. Okay, and you can see that the cholesterol molecules, children, cholesterol molecules are embedded in between. So here is a cholesterol molecules embedded, which is going to reduce the fluidity of your plasma membrane. So this is a way how you can actually check the proteins, membrane proteins, which have been present. So the next thing that I'm going to talk is about the uh, linker proteins. So these are the specific embedded proteins, children, linker proteins. Linker proteins, what they do is, these are the proteins which are attached to your cytoskeleton. See, for instance, if this is going to be your cell, and inside the cell, I told you that you are going to have these linker proteins. So this is a linker protein. What the linker protein is going to do is, children, inside the cytoplasm, we are having these specific proteins which are forming the cytoskeleton. So these are also sort of what proteins only, but they are getting all the proteins here in the cytoplasm. Major of them are getting organized. The proteins are getting organized into what? As a cytoskeleton. So proteins in the cytoplasm, let me write this. So proteins in the cytoplasm organized are organized as cytoskeleton, organized as cytoskeleton. So these cytoskeleton, now what we do is, these linker proteins are very much attached to the are very much attached to your cytoskeleton. So now what happens is your these linker proteins, what they do is they are going to actually can cause your cytoskeleton proteins to move. So your linker proteins are enabling, they are enabling the cytoskeleton proteins to move. So what is this going to help in? For example, if you have seen the case of WBCs, they perform diapedesis, right? And many of the WBCs like macrophages, they are going to produce pseudopodia and they are going to engulf the what you call is the foreign bacteria as well as the dead cell bodies, right? So these movements or the macrophages or pseudopodia is nothing but what they are making cytoplasmic streaming movements, right? They're making some cytoplasmic streaming movements, and they even do a diapedesis where they squeeze out of the capillaries and come out. So cytoplasmic streaming movements are being performed. So all these movements are just because of the movement of the cytoskeleton proteins because of the linker proteins. So linker proteins are directly helping in what? Changing the shape of a cell, changing shape of a cell like your WBCs, changing shape of the cells like your WBCs. So this is the protein which is embedded in the what you call is bilipid layer, which is enabling your cell to change its shape and develop the movements. So this is about your linker proteins, children. And next what we are going to have is we have another type of proteins which are being called as the receptor sites. They are going to act as receptor sites. So children, we all know that our endocrine gland cells, right? The endocrine gland cells are going to produce their specific secretions, which are being called as hormones. And these hormones are going to act only on, they are going to act only on the target cells, right? Only on the target cells they act. How is this possible? Because these target cells are going to have what? They are going to have receptors, because the target cells have specific receptors, specific receptors for a hormone. Okay, that specific hormone, that specific hormone, we are going to have their receptors. So they act only on the target cells. For example, if it is going to be thyroid stimulating hormone, this is going to act only on the thyroid but not any other gland, because they have receptors. The thyroid gland is going to have the receptors for the TSH. So they have the thyroid stimulating hormone receptors here, due to which they are acting only on the thyroid gland. So these are the receptor sites we are going to have. And we all know that, children, there are two different types of hormones that we have. One is going to be the peptide hormones, and the next category that we are going to have is going to be called as a steroid hormones. So if you look at the peptide hormones, they are being made up of amino acids, and the steroid hormones are made up of what? A fat, which is being called as your cholesterol. So imagine if your amino acids, these, see, let me come into this detail. Now this becomes a very interesting topic here about the peptide hormones. Okay, the peptide hormones, and coming on to your steroid hormones. Steroid hormones. Now, children, remember that I told you that every hormone has its own sort of receptors, right? So these peptide hormones are going to have their receptors on the membrane, on the cell membrane. They are going to have their receptor sites. Whereas the steroid hormones are made up of what? Made up of cholesterol. So they are lipid soluble. So they are lipid soluble. Likes attract each other. No, it is unlikes are going to attract each other. But here, if you look at the case here, the lipids are going to move across the lipid soluble tails, right? So directly your steroid hormones are going to move across the cell membrane. So if I'm going to consider this as a steroid hormone, this as a steroid hormone, children. This is your steroid hormone. So since it is lipid soluble, this makes a easy movement across your bilipid layer, and its receptors are being present where, they are being present inside the cytoplasm, or they may be present inside the nucleus. So it depends upon the type of the hormone that we are talking about. So now you can see that this steroid hormone can actually have its receptors here, or it can have the receptors inside the nucleus. So if it is going to have the receptors in the nucleus, children, what it is going to do is it is going to actually initiate, your what we call as transcription of mRNA, that is your DNA transcription, and we are going to produce mRNA, and that specific sort of gene is going to be produced for the functionality of that hormone. So this is going to be the steroid hormones, but the peptide hormones are going to have the receptors on their cell membrane only, because these are not lipid soluble. They are not lipid soluble. And the other important thing that I would like to specify here is steroid hormones can be taken orally as medicines. For example, if I'm going to talk about a birth control pill, a woman can take a estrogen pill. So that can be actually acting, can be absorbed, into your what you call is the cells, into the body, what I call as. But if you're going to look at these peptide hormones, they are digestible. They are digestible, children. So now what happens is, if you take any oral pill of your insulin, because insulin is also a peptide hormone, right? It's an example for peptide hormone. If you take an oral pill, now what happens, insulin is going to be digested into amino acid form. So how can it act on your what you call is a target cells? This can't happen. So orally you can't take a peptide hormone, but orally we can take a steroid hormone, because they are directly absorbed. They are not digested. They are not digested. That's the reason we have no insulin pills, but we have our insulin injections. Okay, so this is an important thing that you need to understand about the receptors. And children, the hormones that are acting here is nothing but these hormones. For example, I told you we have receptors, right? So receptors are going to be the ones which are receiving the hormones. So the hormones, for example, if this is going to be my receptor, so if I'm going to consider this as my receptor, and if it would be my what you call is a hormone coming up into this particular site, and this is going to be my hormone. So what I can call these as terminologies is, this will be my receptor, and someone who is going to activate a receptor is going to be the one that is being called as a ligand. So who is a ligand here? Ligands can be either your hormones, okay, or they can be your neurotransmitters. They can be your neurotransmitters. Remember, children, your cells may have multiple number of receptors. They may have multiple number of receptors. One may be acting for growth hormone. One may be acting for the adrenaline, what we call as epinephrine. One may be acting for the insulin, okay, and the other might be acting for the estrogen. So these are like, we have n number of receptors, that is the proteins which are acting as receptors for a specific sort of hormone, or what you call as a neurotransmitter, or any other chemicals which are going to activate your receptors. Okay, and the next thing that we are going to speak here is about your glycoproteins. So next, lastly, what we are going to have is the other protein which is embedded, called as glycoproteins, children. Glycoproteins are the actually forming the recognition sites for your WBCs. These are the recognition sites, or what we call as identification markers. These are the identification markers for your WBCs, in order to distinguish the self from your foreign cells. Okay, self from the foreign cells. So you have your body has your body cells have your own specific type of glycoproteins that do not match with any other individual, so they will be acting as a recognition sites for whom, for the WBCs, to discriminate or to distinguish between what is self as well as what is a foreign particle. So this topic I have covered in the kidney transplantation when I spoke about the organ transplant rejections, organ transplant rejections. So I spoke about this where I told you that people would like to go for the immunosuppressant drugs. So that I have covered it up detailedly. If you are interested, please log into the video called Kidney Transplantation, where I spoke about the glycoproteins, and I even spoke about glycoproteins in detail in the blood groups video also. So children, I request you all to just go through such type of videos and build up your concepts well. So this is the one what I needed to speak about, children, about the different proteins like glycoproteins, which are acting as what the antigens, surface antigens, or the surface markers. They are identification markers. We have these what we call is the receptors. So the proteins are acting as receptor sites for the different ligands. We have enzymes. We have linker proteins giving up a change in the shape for a your cells, and we are having transport proteins which are transporting either by taking ATP or not by ATP. We have these ion channels which are opened up for a small size particles, and we spoke about the cholesterol which is being present. Now let me show you, children, if I have further few of more others type of slides. Let me come and show you these slides to you all, such that it will be easy for you all now to remember. So I was talking about the active transpiration ion channels. So these are the ion channels that we are having, which are transferring your what we call as a sodium as well as a potassium ions. Okay, and next what I have is the facilitated diffusion that I have spoke about. So these are the proteins which are transferring, they are changing their shapes. These are the carrier proteins. Okay, this is the ion channel once again, which is going to, but here I'm speaking about the glucose molecules getting transported without any ATP, okay? And we have the other carrier proteins which change their shapes accordingly and move the materials or molecules across your cell membrane. And coming on to children, this is the plasma membrane there, where we could see the cholesterol, the protein channels, ion channels that we have seen, the peripheral proteins. These are the cytoskeleton that you can see here, and the glycoproteins which act as your antigens or the surface markers, and we are having these glycolipids here and the cholesterol embedded in between. And let me show you the different types of the proteins that I spoke to you all now. So these are the embedded plasma, that is the proteins which are being present, like your ion channels which open up togetherly from top to the bottom of the bilipid layer, allowing the easy diffusion. And we have few of the enzymes which are going to act for the catalysis process, biocatalysts they are. And we have the receptors, receptor sites, the proteins are acting as receptors. We are having the glycoproteins. So this is your sugar chain with a protein, so glycoprotein. And a few of the proteins are helping in cell adhesion processes. And I spoke about the linker protein attached to your cytoskeleton, by which they are going to help in the movement of the cytoskeleton, changing the shape of the cell. So children, if you have liked this video, please do hit the like button and try to comment in the comment section how you like the video, and try to subscribe my channel and share with others too. Thank you all. Thank you.